Mr. Anthony, maybe we can make it like YouTube live streaming.
Okay, sure.
Yeah. Hope Kevin's laptop is fixed already. Shall we wait for another 2 more minutes? Maybe the participants are joining. All is just a few minutes. We are trying to fix the YouTube live streaming, so please bear with us. Yes. Now it works. Perhaps we can start the meeting first. Bear with us for a minute. It's good time to start the program, if I'm not mistaken. Good morning to most of you, and good evening to some of you joining from United States and other countries. Thank you for joining this International Symposium on Horseshoe Crab Conservation. It's in line with, in conjunction with International Horseshoe Crab Day second year. This program is organized by International Islamic University Malaysia, co-hosted by IPB University Indonesia, and fully supported by IUCN Species Survival Commission on Horseshoe Crab and the Beibu Gulf University, China. Thanks for the presenters. I'm sure there is no introduction needed for the presenters because they are the co-chairs and the members of the IUCN SSC and so on. For some of you who do not know the first presenter, Professor Mark Barton, he is currently a co-chair of IUCN SSE Species Survival Commission on Horseshoe Crab, and he is the professor in biology from Fort Ham University, United States. I would like to introduce about this program. This program, as I said, it is in conjunction with the second International Horseshoe Crab Day. So each presenter will present about 25 minutes max, followed by a question and answer session. Of course, two presenters will present. The first two presenters will present first, followed by the question and answer session, and same way goes to the third and fourth presenters, and fifth and sixth presenters. Please bear with us if there is any technical glitch. And I would like to pass this floor to Prof. Barton and to introduce and to some of you, he, by his name, a species of the horseshoe crab was named, especially the species which is identified from the Scotland, the fossil record named after him. So this itself is enough to introduce himself. And now I pass the floor to Prof. Mark Barton for his first presentation.
Thank you, Dr. John. Can everybody hear me okay?
It's perfectly okay, Prof. Please.
Yeah, okay. So if I can share screen, we can get started.
Prof, we don't hear your voice. Is it muted?
He has muted.
Prof, perhaps you have to unmute yourself, Prof. Sorry.
We should be okay now?
It's perfectly okay, Prof.
Okay, sorry. I'll just jump right in then. So horseshoe crab research has a long history in both the Asian countries and in North America. But at least initially, there was relatively little international collaboration, in part because of language difficulties and just the difficulty in the pre-internet era of communicating with colleagues across the oceans. And I want to acknowledge just briefly some of the people who made major contributions to the horseshoe crab science in different parts of the world. These are names and faces that will be familiar to many of you. So Huilian He and Changpo Chen in Taiwan, Xu Hang in China. In Japan, there's a long history of horseshoe crab research. Dr. Sakaguchi was really the dean of horseshoe crab biologists in the latter part of the 20th century. He had a number of very prominent students, including Tomio Ito and Hiroki Sugita. Dr. Sakaguchi made major contributions to developmental biology, anatomy, physiology, many other aspects of horseshoe crab biology. And his work was extremely influential in Japan. In the US, again, most of you will recognize the names Carl Schuster and Bob Barlow. Carl made a tremendous number of contributions in anatomy, population biology, conservation of horseshoe crabs over his long and distinguished career. Dr. Barlow really was a neurophysiologist working with horseshoe crab vision, primarily made some significant contributions in that area, and also expanded his studies into behavior. Perhaps the leading biologist in terms of horseshoe crab behavior in the US is Jane Brockman, shown here at the mudflats in Beihai a couple of years ago. Jack Levin, Tom Nowitzki, two of the pioneers of the work with LAL, Limulus amoebocyte lysate, and Bob Loveland, my longtime collaborator, who we've worked with together since the mid-1980s on various aspects of population biology, primarily in Delaware Bay. There's also a long and distinguished history of horseshoe crab research in India, and several of the people, again, are well known to you: Anil Chatterjee, Vasudha Tripathi, and Jay Mishra, all made major contributions to horseshoe crab biology in India. But at least initially, people in the different countries were working somewhat in isolation from one another. And that began to change in the late 1980s, when Dr. Sakaguchi and four of his students visited us in Delaware Bay. And this really sort of cemented the friendship between US researchers and Dr. Sakaguchi's group. In 1994, Carl Schuster and I spent about 3.5 weeks in southern Japan with the great help of Dr. Sakaguchi, Dr. Sugita, and many others. Took us around to some of the breeding sites in Japan, to museum collections, and so forth. And this is a poster from lectures that Carl and I gave. There's my name down here, M something or other, CN Schuster, and we actually participated, of course, with translators in a number of conferences and sessions in Japan. Carl shown here examining Carcinoscorpius at the Shimoda Marine Lab. Here's Carl with Dr. Sugita, Dr. Ito, and Dr. Sakaguchi after one of Carl's lectures. Picture I particularly like is that of Dr. Sakaguchi and Carl in front of the entranceway to the Kabutogani or Horseshoe Crab Museum in Kasaoka City, Japan. Another important influence in Asia was that of Brian Morton. Dr. Morton at the time was editor of a very prestigious journal, Marine Pollution Bulletin, and he wrote this short editorial in 1999, which really sort of opened my eyes to the widespread problem of the decimation, depletion of horseshoe crabs, not only in Hong Kong, but elsewhere in Asia. And Dr. Morton was also, as I understand it, the major influence that got my colleague Paul Shin to pick up horseshoe crabs as an area of research. In 2003, Carl, along with Bob Barlow and Jane Brockman, published this well-known book now, The American Horseshoe Crab, which, despite its name, had contributions from Dr. Sakaguchi and other Asian scientists. So it was really an international effort that focused a lot of attention on horseshoe crab science. Things really began to gel in terms of international cooperation with the first of the symposia that was held at Dowling College in June 2007. This was the first of 4 similar meetings in other locations. But this was really the first time that many of us had a chance to meet one another face to face. Up to this time, people had sort of known of one another's work through the literature, but it was certainly the first time that many of us got to meet and discuss horseshoe crab science. There were about 125 or so participants at that first conference. And in 2011, Dr. Shin, I believe that's him over there, organized the second workshop, which we focused on Asian horseshoe crabs. That was held just a little over 10 years ago now in Hong Kong. And among the outcomes from this meeting was a sense that we needed to take action for the conservation of Asian horseshoe crabs in particular. It's well and good to give talks and symposia, but the consensus was clearly that Asian horseshoe crabs were imperiled, and the best vehicle to try to stem the tide of the decline of those animals was through the establishment of a specialist group within the IUCN, or International Union for Conservation of Nature. Now, many of you are part of this organization, and the next few slides will probably seem a bit repetitive to those of you who are familiar with IUCN, but just as a brief review, IUCN has within it a number of programs, one of which that we primarily deal with is what's called the Species Survival Commission. And there are dozens of similar groups, anything from plants to mammals, fishes, spiders, many different taxa have specialist groups. There are other programs within IUCN that I'm not going to discuss, but again, it's a very large, multifaceted organization. Now, the aspect that I want to talk about and leads most directly to conservation issues is what's called the IUCN Red List. And if you land here on the home page for the IUCN Red List, you see that you can enter the name of a particular species, and that then brings you to information and reports about the status of that species. Now, IUCN Red List has a number of different categories where a species might be placed. So there are some taxa which are just not evaluated. In fact, many species for which there's no reporting at all yet. Then for those that are evaluated, in some cases, the data are inadequate to make a proper judgment, of the judgment of where it might belong on this extinction risk hierarchy. Where data are adequate, if a species has a very large population and it's not seen to be in any danger at all, it would go in the category of least concern. Somewhat more risky status would be near threatened, and then vulnerable, endangered, critically endangered. And then where you don't want to be is up at the top, where a species is deemed to be either extinct or existing only in captive situations that would be extinct in the wild. So IUCN red listing is an important tool for conservation. Now, when we began as the Horseshoe Crab Specialist Group in 2012, all three of the Asian horseshoe crab species were listed as data deficient. It's important to point out that red listing is a tool. It's not the end point, but really a tool in conservation. A species listing on the red list doesn't guarantee that it's going to be protected, but it does give greater credibility to conservation efforts at the national level. We were organized as a specialist group in 2012. We put in a proposal following the conference in Hong Kong. And as we are currently comprised, we have two co-chairs reflecting the geographic distribution of horseshoe crabs. So Paul and myself are the current co-chairs of the group. We have a very active and vibrant steering committee composed of scientists from both Asia and North America, and a number of other scientists and conservationists are members of the group. So we have approximately 89 total members at this time. And I'll talk a little bit more about the breakdown of that membership a little bit later in my talk. The organization of our specialist group is reflected in this diagram. So at the moment, we have 5 active working groups, a group led by Kevin Laurie, who you'll hear from shortly, on Asian horseshoe crab red listing, the limulus red listing process, which is led by Dave Smith, trade, industry, and use, led by Glenn Gawvry, education and public awareness by Jennifer Matei and Billy Kwan. And then our newest group, these first 4 were formed at the initiation of our specialist group. But in 2019, we decided to add a 5th working group here, the Asian horseshoe crab group, which is led by Billy Kwan and has a number of other members, as these all are comprised of a number of these different specialists. So this is how our group is organized. They all report to the co-chairs and steering committee, and we have regular Zoom meetings of the steering committee to get reports from all 5 of these working groups. Now, where does a species end up on the red list? And that's a challenge that the working groups face. IUCN provides very detailed criteria for whether a species belongs in a particular category. So whether something should be vulnerable or near threatened or endangered or critically endangered, etc. There are very specific guidelines which IUCN provides to us. The process begins with a working group that reviews all available evidence. They then compile a draft report sent to the within the specialist group. We have a group of specialists who review those reports, and the revised red list assessment, once it goes back from the SSC, from the specialist group chairs back to the working group, hash out any differences. That then goes to IUCN, Species Survival Commission, for further review and hopeful approval. And Paul and Kevin will give more specific details vis-a-vis the Asian red listing. So what we've achieved is thus far 2 out of the 4 red lists have been revised. The American horseshoe crab, which was formerly least concern, was upgraded to vulnerable in 2016. And the tri-spine horseshoe crab, formerly data deficient, is now listed as endangered. And the red list reports for the other 2 species are in progress. I want to talk a little bit more about some of the other work of the specialist group. Red listing is part of what we do, but it's not the only thing. And our IUCN always asks us to look to set targets for each 4-year cycle. The 4-year cycle now is going to end in 2024. So Paul and I, in conjunction with the other members of the steering committee, developed a number of targets, which we then communicate to IUCN. So one of these targets is for us to develop best practices for the assessment of horseshoe crab populations. A lot of people are doing this sort of work both in North America and Asia, but there's no standard method yet, and people are doing things differently, and it would certainly be beneficial for our efforts if we had some degree of uniformity in how the populations were sampled in different locations. So this was a target that we initially set for the prior 4-year period. We've made some headway on that in terms of compiling a list of monitoring practices from different countries. So this is still an ongoing goal. Another goal that we set was to establish the Asian Horseshoe Crab Observation Network, which was spearheaded by the Asian Horseshoe Crab Working Group. And actually, this is the first year this program has actually gotten off the ground. It's being done primarily in China, and Billy Kwan has led that effort to start this up, and hopefully can diversify to countries outside of China in years to come. I mentioned before the red list process. One of our goals is to get Carcinoscorpius, the mangrove horseshoe crab, and the coastal horseshoe crab, Tachypleus gigas, get them out from this no man's land of data deficient into one of the accepted risk categories. Kevin will talk more about this later on, so I will defer to him on that. But that's one of our priorities. Another priority is to complete the green list assessment for Tachypleus tridentatus. For those of you who are not familiar, the green list is basically an outline of a recovery plan for the species. So we know that tridentatus is endangered, but what steps could be taken that would perhaps bring back the numbers to some prior larger population size. The green list assessment for Limulus, the American horseshoe crab, has already been completed, but we'd like now to move on to do tridentatus. Another important goal that we set in our communication area was to bring about a publication of some of the papers that were presented at the prior workshops. That's been achieved. The book is in press. John Tancredi is the lead editor on that. We, of course, are involved with helping to coordinate activities in celebration of the International Horseshoe Crab Day That started last year, and this symposium that Dr. John has kindly organized is a part of that effort. We'd also like to continue meeting in person with some future dates. We know we have a special session coming up at the November Coastal and Estuarine Research Federation Conference, which is going to be virtual. And I know a number of you are interested in knowing what the status will be of our 5th international workshop. We had been holding these at 4-year intervals, obviously, until we have greater clarity with respect to the end of the COVID-19 pandemic. We are sort of putting the next international workshop on hold. Hopefully by 24 we can do it, but it's certainly something we would like to be able to get back to. Our specialist group membership, I just want to very briefly mention, we have, as I said, about 89 total members. We are about 2/3 male. About 2/3 of the representatives are from countries in Asia, with most of the remainder being from North America. US, China, India, Japan, Hong Kong, Singapore, Malaysia have the largest number of members. We have some countries where we have only 1 or 2 members. The reason I mention this at all is that the IUCN Species Survival Commission is actively encouraging greater diversity within the specialist groups, not picking on us in particular, but just generally, IUCN is interested in promoting greater diversity with respect to gender, nationality, and looking to get more early career scientists and people who are not necessarily affiliated with academia. So we're looking to broaden our base of people in the specialist group. The last thing I just want to mention comes back to this point about international cooperation in terms of publication. This was an interesting paper published in Frontiers in Marine Science a little more than a year ago, which basically looked at the degree to which co-authorships occur between different countries. And what you can see here is there are several, the size of the circle is effectively the number of publications about horseshoe crabs coming from different countries. And then the linkages indicate where there's cooperation as meant by co-authorship. And so US, Japan, and China, the largest 3, Singapore, Malaysia, India, follow. But what is important to see, and again, the details are not important, is the degree to which there is co-authorship between scientists of different countries. And this was something that wasn't really seen, 30, 40 years ago. So this is a very positive development. So in summary, the IUCN Specialist Group was formed in 2012 as a direct outcome from the meeting in Hong Kong, where everyone was quite clear on the degree of trouble that horseshoe crabs, particularly those in Asia, were facing. Red list process, as I mentioned, is a part of what we do. It's not the entirety of what we do, but it's a major effort right now to complete the red list assessment for Tachypleus gigas and Carcinoscorpius. And finally, in closing, we are hoping to be able to see everyone again in person. This was our last meeting, and hopefully events such as what we're doing today can lead to further international cooperation in horseshoe crab research and education. So I thank you, and I'll turn it back to Dr. John.
Thank you, Prof. Thank you for your presentation. Because of the time constraint, because many people are joining from other places as well, I mean, with different time zone, I would like to move on to the second presenter, Professor Paul Shen. He is, of course, a co-chair for the IUCN SSC on horseshoe crab, and he was the professor from City University of Hong Kong. Now he is joining us from Canada. Thank you, Prof. Floor is yours.
Okay, thank you, Dr. John, for organizing this online meeting to celebrate the second International Day of Horseshoe Crab. Mark has already given you a very detailed development and the work of the IUCN specialist group. In my talk, I'm trying to share a few ideas of how we could be able to move forward in terms of the conservation and research of horseshoe crab in Asia. A part of the material that I'm going to talk about probably have to have included in my last talk at the China meeting in 2019. However, I think that I will need a bit more information in terms of research area. Let me start with a recent expert questionnaire survey conducted in 2019 and during the China meeting in June of the same year. Results are based on responses from 46 participants from 11 countries and regions. This graph actually reaffirms that socio-economic developments, particularly in the coastal habitats, is the major threat to Asian horseshoe crabs in general. The majority of the respondents in the same survey also regarded that the management habitat and the involvement of public awareness and participation will have to be regarded high on the agenda for our future conservation work. On the left side, the respondents also highlighted that collection of baseline data and the identification and evaluation of habitat and management will also be important if we want to move forward in this area. So I'm going to share 3 conservation priorities, which I think will be very important for us in the coming future. The first one, of course, as like Mark said, that is to acquire internationally and nationally recognized status for our horseshoe crabs so that they could be able to be under certain legal protection. I think a recent success story is the listing of the transparent horseshoe crab in China. Now it is a second priority protection species nationally. based on our assessment from the IUCN web list result. I'm going to talk about a bit more in the following slide about the IUCN green status species. That's what Mark mentioned, the green list assessment in a following slide. For CITES, I think everybody know about CITES. And of course, if we could be able to place horseshoe crab on one of the appendices of CITES, then there may be a possibility to help control the harvest and cross-border trade of horseshoe crab for food consumption or for other uses. This concept of World Heritage Species is very similar to existing World Heritage Sites. This is still a concept, and there's no species designated formally under this category yet. But this proposal is to recognize that certain species actually play a very significant role in our culture and heritage and natural heritage. So this kind of species should warrant further global protection. There's one idea that that we could explore in the future. Now, for the BSEP, or the National Biodiversity Strategy and Action Plan, I mentioned that in my 2019 talk, and I have a slide to show a bit more details and how it would benefit for the conservation of horseshoe crab. Now, for the IUCN green status list, in previously they named it as green list assessment, but now they changed it into green status of species. Actually, it's a very new concept to assess a particular species in terms of how they revive or recover based on either past, that means that we accepted the past as well as the future interventions in our conservation efforts. And IUCN is planned to incorporate the green status species with the wet list assessment process in future. So that means in future, definitely both will go together as a more comprehensive assessment for a particular species. For creating or assessing green status of species, this is based on a generation of a green score. I'm going not to tell about the details, how about the score being generated, but basically it's based on three areas of recovery listed on the slide. Whether a species fully recover, if it's present in all parts of its range, whether it's viable, that means not threatened by extinction in all parts of the range, whether it's performing its ecological function in all parts of the range. So there's a specific detailed guidelines, criteria, and background information that can be accessed from the IUCN website. As Mark said, that we have already done the green status of species assessment for limulus, and we have just formed a team comprising members of our specialist group from 7 countries or regions to embark working on the green status of species assessment for the transparent horseshoe crab. And hopefully we could be able to move forward with this task in the coming year. The BSEP or the NBSEP, the National Biodiversity Action Plan, actually is a requirement under the Convention of Biological Diversity. It's a very important kind of process What it really means is that that we will ask the government or the authority to integrate conservation and sustainable use of biological resources in any or every national economy and policy making process. That means take the conservation and the sustainable use of biological resources as a major, one of the important issues, not just regarding economic returns, regarding GDP, GNP, etc. One has to also include the importance of biological resources. To me, this is a really good opportunity if we could be able to be involved in such process in the developing this. I and a couple of us, like Edgy and Kevin, were fortunate to be involved in the development of the biodiversity strategic action plan in Hong Kong, and we could be able to put horseshoe crab as one of the important conservation species within the plan, and that means the government will have to add on the actions listed in the plan. And I think one of the suggestions is to put horseshoe crab on the local vet list created in Hong Kong. So I think that quite many countries already have done their BSEP, but that is a routine process. That means after several years, then you have to develop or revise the plan to suit the particular situation. The second priority for conservation of Asian horseshoe crab, definitely, of course, is to protect the habitat where horseshoe crabs are thriving. This is an in situ conservation approach. And of course, you can do it via enhancement of already degraded habitat through restoration or revegetation of mangroves, seagrass, marshes, etc. Or you can establish or try to enhance management in existing reserve or parks. Because as we know that most of these parks or reserve ecosystem like mangroves or marshes, which can provide a vital role also in protecting horseshoe crab habitats. The left picture shows the Guangxi Bihai Coastal Wetland Reserve. That's the one that we visited at our 2019 meeting in China. And the right picture shows the Mandai mangrove and muskrat in Singapore, which will be open in 2022, next year. In fact, this is an important habitat for the mangrove horseshoe crab. So you can see that once there's established management protocol for such a reserve or park, then there would be a guarantee for certain protection for the horseshoe crab as well. They are associated with the reserve and parks. To create or establish MPA or marine protection area specifically for horseshoe crab, that usually requires a lot of effort, including collection of scientific knowledge and data. The figure shows our attempt in Hong Kong to propose 3 MPAs, one on the northeast and 2 on the west. This is the Deep Bay Horseshoe Crab Protection Area, and Park Line. I think that this area that some of you may have visited there at the in the 2011 workshop, and the one down here close to the international airport. When we propose such kind of MPAs, the collection of data or investigation in terms of species density distribution, core analysis, determining the extent of foraging area are very important. Of course, we have also to include consideration with the population genetics. I think that Professor Jeremy heard later, in his later talk, we will present more about this aspect. Of course, the third priority for conservation is to raise public involvement, as you can reflect it from the responses of the public surveys, the expert questionnaire survey. This could be achieved through partnership with local community, NGOs, or conservation groups. I'm aware that there are many such activities being organized in many countries in Asia. The left picture shows local fishermen in Malaysia try to clean up their shores. Okay, so you can see that the shore could have been enhanced in terms of conditions so that the mangrove horseshoe crab can be able to live in the area. The right photos are from Indonesia, and you can see that a partnership with the local fishermen and even their family member trying to release the entangled horseshoe crab from their bycatch in their fishery operation. Mostly could be catching of swimming crabs in inshore waters, and then the healthy or the live horseshoe crab will eventually return to the water. And there are a couple of papers in the coming horseshoe crab book, as mentioned by Mark, citing or presenting result and observation of bycatch data of horseshoe crab. This could be of interest to read when the book is out. These two, the Ocean Park or OPCF juvenile horseshoe crab very program is a very successful case, and I have the privilege to in working with the Ocean Park in this project, and I think that Edgy, my colleague, would give you a more detail and experiences to learn from such project. And we have started project since 2009. The right picture shows that how school children try to study and rear the juvenile horseshoe crab in the schools, and then they also involved in the beach cleaning to enhance the habitat condition for horseshoe crab in Hong Kong. The other program, which I think is very useful, the logo missed here, but the web page address is down here. This is the Project Nimbus, led by Professor Jennifer Mattei in the USA. This is a program based on citizen science to engage the public to study horseshoe crab ecology, as well as to educate them in terms of knowing how horseshoe crab are related to the ecosystem and also related to human health. So you can find more details from this web page. If I turn into a research area, you can see from remember from the survey that population baselines are important. And for and that Mark has already mentioned that we are trying to come up with certain streamline or standard protocol for monitoring populations. But I think quite a lot of work has been done in China or in Hong Kong in particular, or even in Malaysia, as I heard from Farida the other day, that survey of Asian horseshoe crab particularly at the nursery site has been established. This slide shows about a proposed standard kind of protocol for monitoring the juvenile population at their local nursery site by using transect as well as quadrat. Of course, the number of transect, the number of quadrat used had to depend on the specific site location. And that's why really within the Asian horseshoe crab working group has established the horseshoe crab observation network program, as Mark introduced earlier. I think the aim is to really set up to adopt a standard protocol, survey protocol, field guide, and data sheet, so that we could be able to provide similar information for horseshoe crab population information, etc., across a lot of the geographic range and their nursery habitats. The program also offers technical support and on-site training for those participants. And most of the participants, in fact, from the partnership that Billy has secure from government agencies, universities, research institutes, and conservation organizations. As far as I know, today, in fact, Billy just finished on-site training in Mainland Island a couple of days ago, and there's the field training occasion kind of pictures. At the moment now, there are 13 organizations involved in this program with 17 monitoring sites, primarily in China, including Hong Kong. There's the plan to expand this program to other Asian countries next year and onwards, depending on the resource and other considerations, for example, travel, etc. For adult population, I think that is the problematic kind of area, and frankly, I don't have a lot of experience in collecting adult horseshoe crab in Hong Kong. One of the ideas is to apply this systematic bottom trawling to estimate population size. This is of course ideal, but could be resource demanding and logistically challenging. And I guess that in doing so, you have to have cooperation with government agencies and or research institutes who have the capability and resource to do that. But I think that is something we can think about in future. Maybe we can also involve fishermen organizations to help when they are doing their fishing operations in the coastal waters of the area where horseshoe crab may be found. For passive approach in collecting population data, I think the well-established method is the mark recapture. Just like we take the horseshoe crab, release them, and hopefully we can we collect them back and estimate the population extent. The other possibility is to use bycatch data. And I found it pretty interesting when I reviewed several papers in this area. If we could be able to plan well with the local fishermen how to record the data, the cache data, and then in a systematic way, one may be able to use the data to estimate the population size. Something I think that is really worth to explore future, particularly in area where bycatch is a problem, like in Indonesia and in Malaysia. This is my favorite area. One of the favorite area is to explore the feeding and food web ecology to review the utilization of food resources by horseshoe crab within the habitat, whether it's on nursery ground or maybe in future in their feeding ground. The left panel shows a relationship between the use of stable isotope carbon and the stable nitrogen value. You can see that there's a positive relationship with the value with the increase in the postosomal growth of the horseshoe crab. And then the most of interest, of course, is to look at how the horseshoe crab utilize the food resources available to them at the habitat. This graph shows that this byproduct of the two stable carbon and stable nitrogen isotope values reveal that cork, grass, and microalgae, okay, and or the mixture of both would be the major food source for this juvenile horseshoe crab collected in Guangxi by Billy from different sampling sites. The other use of the stable isotope data is to estimate contribution of food sources of horseshoe crab based on the isotope basic model. This all available now, and you don't need me to create a model yourself. And this is a study that Man Hong from Shanghai did to estimate what are the major food sources for the juvenile horseshoe crab in Guangxi, China. And you can see there are three different size group, and you can see that bawas are the major food items. However, you can see that when the horseshoe crab grow larger, and then they also shift into a mixed diet mode with bawas and crustacean. So this is a pretty useful information if you want to know more how horseshoe crab utilize the available food sources at a particular area. This is also important because of the declining horseshoe crab population in the wild, and the techniques and operating procedure for artificial breeding and larval and juvenile culture of horseshoe crab. I think it's well studied in China or Hong Kong, as well as in Malaysia. And then the further told me that there is a culture center for the breeding and culture of the juveniles there. So I'm not going through the detail. You can see that there's a paper from Frontiers in Marine Science that shows how would be the optimal spawning, incubation, and juvenile rearing procedures. And in fact, I would recommend you to read a couple more papers from this journal, this issue, okay? Because there is a special section on aquaculture and rearing of horseshoe crab, not just in China but in other areas, and including in USA. So they're all open access, so you can download and you can read and all this if you have time. I also know that in fact the associate of Cape Cod company, and this is a company producing LAL in USA. They do have a project called Horseshoe Crab Sustainability Project, and you can find more detail on this link. And they have a patent pending process to incubate juveniles. And then I think a couple of their representatives, they attend the China meeting in 2019, and they are willing, or they will be happy to collaborate with anybody in Asia if they would like to use their process to culture horseshoe crabs. And I think that Dr. John has the collaboration with them, and so you can ask him for more information or for contact. Billy also recently have a patent ecological simulated culture system, and the details are in Chinese, so I'm not going to bother you to read all this day. I'm happy to have a picture from him about how the system works. So if you have interest, and then you can approach Billy, and probably he will send you more details, translate the patent details from Chinese to English. It's a system to culture, to raise and culture juvenile horseshoe crab, particularly the transparent horseshoe crab. Now, once we have the horseshoe crab, juvenile horseshoe crab released to the field, and then of course you need to trap them. But before you release them, there are also a lot of problems with captivity-related stresses and issues. We have been doing some work on nutrient supplement, use of artificial feed, and then the Professor Shi and Professor Champel has employed a polyculture of juveniles with a baby snail, which has the economic value in outdoor tanks to improve their survivorship as well as their growth. So there's a few papers as quoted here. You could be able to do more, get more details. You want to go into this area. But I think it's very important if we can have a more healthy juveniles before you release it to the field. And once you release to the field, of course, you need to monitor them, to follow them. One of the methods is to use this internal PIT, passive integrated transponder. And then you can inject this one into juvenile horseshoe crab. Of course, you won't be a so small one. Probably it will be 6 or 7 install. And then you can detect it by using a scanner, because each of this PIT has the specific code. So and then we have tried this out in Hong Kong, and so there's always a kind of a potential to use this method, and this method is actually employed to fishery migratory studies for a long time. The other area which I don't know a lot, but I think is important, is the habitat evaluation. One have to establish a scheme, mostly just as the one I quote here. You can get more detail how that will be done. Usually you need to do it with GIS or mapping exercise. And then there are also a protocol or approach using rapid assessment methodology. And then in this, more similar, but have identified certain indicators like human pressure on particular habitat, introducing of pollution in the area, how the living resources are exploited, how protection of coastal area is done, and also like the examination of biodiversity abundance and the health of particular species. So I think that's all available from existing literature. That's this area that hopefully we can do more on this part. Frankly, I haven't done a lot in this area, but I think that's very important, judging from the responses from the questionnaire survey. I want to spend one slide on capacity building and funding opportunity, because I think they're very important, particularly for country in Asia like us, that we may not have all the resources. There's this first two, the conservation leadership program at the edge of existing program may be useful. I'm not saying that must be applicable to horseshoe crab conservation, but may be useful. Particularly this one, CLP, is support high priority diversity conservation through building leadership. skill, particularly for early career conservationists. And I'm happy to report that, in fact, this year, CLP has awarded a funding to support a program or project in Indonesia to study horseshoe crab in association with community involvement. And then, in fact, the Asian Horseshoe Crabs Working Group, particularly Billy, will act as an advisor to the project. So we look forward to having more results from the project when you start this year. This edge, what edge mean? Edge means the evolutionarily distinct and globally endangered species. Endangered, okay? So there's a list of this such kind of edge species on the website, and also based on IUCN or other sources. So if the species is listed on the particular program, and then they will offer you a fellowship for future conservation leaders to work on that particular species. They may not directly apply to horseshoe crab now, because I look at the list, mostly would be mammals, the plants, and the invertebrate group includes just the corals. But that may change. I don't know, but that is something worth to explore. Of course, we have the IUCN SSC internal grant. This one, of course, support activity specified by specialist group in their annual targets. And we were lucky in 2019 to have the support from this grant, so that we can fund participants from your country to attend the meeting in China. Now I'm aware that the current applications for the grant is open. So if you guys have any ideas, you may have to approach us so that we could be able to generate a kind of proposal. But mind you, the proposal had to follow or the SSC planning cycle. That means, as Mark has mentioned, that is the network access at this and they communicate this kind of cycle and this thing. So if you are interested in doing so, you have to come to us, and then we will see whether we could be able to help. I think the application deadline is sometime in July, so it's not long for the submission date. Conclude my talk, I think is that I would encourage everyone to work together, particularly those who are horseshoe crab lovers, because I guess every effort counts in the conservation of horseshoe crab in Asia. Just try to initiate your idea. You look for collaboration, whether it's the local, regional, national, or international. I think that could be feasible. And then we could establish a network, share our outcomes, and eventually we can consolidate our experiences, and then we can move forward in the conservation of horseshoe crab in Asia. And this artist's impression from India shows how horseshoe crab is symbolized on the beach in Beihai. Last but not least, I would like to dedicate this talk to two persons. First, like Mark said, is Professor Brian Morton. Brian was the founder, who just passed away a couple of months ago. Brian was the founder of marine biology and conservation in Hong Kong, and he was the first to study horseshoe crab as well. SG and I have benefited from his advice and his teaching as well, so that we can follow his steps to study and conserve these amazing animals in Hong Kong. Let us not to forget or let us to remember Professor Jayan Mishra. He from India. He was a long-time researcher in horseshoe crab and also a long-time member of our specialist group. Unfortunately, he passed away due to COVID. So I hope that both of them they will be sorely missed by all of us, and I appreciate all their work. So thanks for your attention.
Thank you, Professor Paulchen. I would like to open the floor for a question. Maybe few questions are allowed from the floor. If anyone wants to ask a question, please unmute yourself and state your name and your country. Please proceed with the question. Before we move on to the third presenter, Professor Peter, he's ready with us. If there is any question, please. Okay, I consider there is no question, but I'll just update on the associates of Capcod rearing system. Right now, we since I have a collaboration with the associate Capcod, if anyone wants from Asian country, if anyone wants the same system to be established in their institute, please, you can contact me directly. So the system works well with the Asian horseshoe crab. It is well reared, and we have also sea ranching programs before, and even since last year. So without further, I would like to move on to the third presenter, Professor Peter Armstrong, and he is the Professor Emeritus and from the University of California, Davis, from the Department of Molecular and Cellular Biology. He has an experience of more than 40 years on the horseshoe crab research, and now he is joining us from United States. Please, floor is yours, Prof.
Okay, well, thank you, Professor Akbar John, for organizing the this symposium. And if you remember, you are going to show my PowerPoint slides.
Perfect. Here it is.
Right. Okay, so the, whoops, what happened?
Perfect.
Okay, there we are. So the basic, one of the basic demands made by Darwin's theory of evolution is that the most important duty of every organism is to transmit the genes to the next generation. One of the major impediments to this transfer is premature death due to infectious disease by parasites. And could I have the next slide, please? And the defense that the organism, as it grows up, is the immune system. The immune system having several components. The one that I'll be talking about is the one at the very bottom, the innate immune system. This is the immune system of all invertebrate animals. Next slide, please. And there's multiple strategies for immune defense against pathogenic attack. And I've worked in basically all of these systems of all of these strategies in horseshoe crab. Next slide, please. I'm particularly interested in immune defense systems in long-lived animals. Of course, and horseshoe crabs is usually the starting point for my analysis from my laboratory. Horseshoe crabs, of course, live to be maybe as old as 25 years. They have maybe 10 to 12 years of juvenile life when they can't reproduce, and they live in a very septic environment. So they need a robust immune system to allow them to grow up, get old enough, become sexually mature, And then on that happy day that they're able to reproduce, they have fulfilled Darwin's requirement to transfer your genes into the next generation. I've also worked with lobster, and I'll be showing you some of that research today, and also with humans. This is me and my wife, Margaret, who did much of the research that I'll be telling you about. Next slide, please. I've worked with particular interest on evolutionary conserved defense mechanisms. My laboratory was the first to show that thiolester protein family that had been initially and characterized in vertebrates is also found in other vertebrate taxa, and in this case it was horseshoe crab. And since then, there's been developed a reasonably robust cottage industry in identifying and characterizing thiolester proteins in a diverse array of invertebrates. The C-reactive proteins were initially discovered in horseshoe crab by others. The picture here is of the horseshoe crab serum amyloid protein, and it's this lovely donut, double stacked donut. What I'm going to talk about today, though, is this third illustration, the role of the extracellular blood clot in immunity. The blood clotting systems of horseshoe crab, of lobster, and of human are not strictly evolutionary conserved because they use different families of proteins to produce an extracellular blood clot. In vertebrates, it's the protein fibrin. In horseshoe crabs, it's the protein coagulin. And in lobsters, it's the serum lipoprotein. Next slide, please. And functions of the blood clot are several. What I'm interested in particular are the immune functions of the blood clot, extracellular blood clot. The blood clot is able to entrap invading microbes, prevent their systemic dissemination. And what I'll particularly be talking about today is the ability of the extracellular blood clot to capture microbial toxins. Next slide, please. Next slide, please. Oh yeah, the clotting model in horseshoe crab that I've been using is to collect a small volume of horseshoe crab blood into a petri dish or another sterile surface, allow the cells to attach to the surface, during which time they typically degranulate and release the proteins and the clotting system, and produce a extracellular blood clot, in this case, stained green with antibodies against coagulin, the protein that makes up the clot. Next slide, please. And the microbial toxin that I want to concentrate on in today's talk is lipopolysaccharide, also known as endotoxin. It's an essential component of gram-negative bacteria, principal toxin of gram-negative bacteria, and is an important target of the innate immune system in identifying that gram-negative bacteria are present in the system. That would be true for humans, be true for horseshoe crabs. Next slide, please. And this is just a picture of the membrane system of the gram-negative bacterium. Two plasma membranes, the outer leaflet of the outer plasma membrane made up entirely of lipopolysaccharide, also endotoxin. Next slide, please. And here are pictures of that molecule, lipopolysaccharide. It's next slide, please. Yeah. It's really a big deal. Here, the mortality from gram-negative bacterial sepsis is very high, 35 to 50% mortality, approximately 750,000 cases a year in the United States, a major health threat. And there used to be a dedicated journal that's now the Journal of Innate Research, and there's an International Endotoxin and Innate Immunity Society. So endotoxin, a major cause of human mortality and death. And could I have the next slide, please? So next slide, yeah. So the mechanisms by which multicellular animals can cope with endotoxin poisoning are multiple. In horseshoe crabs, we already know of neutralization by LALF, here toward the bottom, a small cationic protein, and activation of the blood clotting system of horseshoe crabs by endotoxin. Next slide, please. And what I want to tell you today is the thesis that the extracellular blood clot of multiple taxa of long-lived animals functions to neutralize and sequester this major bacterial toxin LPS by the extracellular blood clot. And what is the evidence for this suggestion? Firstly, labeled LPS, I've shown, binds to the fibers of the clot. Fluorescinated polymyxin B, which decorates the clot fibers when LPS is present, fails to bind to the clot when it's absent. And then quantitation of LPS binding. Next slide, please. So here is the capture of fluorescein labeled LPS by the coagulant blood clot of the horseshoe crab. And you can see that the green labeled LPS co-localizes with the fibers of the extracellular blood clot in that clotting model that I showed you earlier, stained with anticoagulation, coagulant being the protein that makes up the fabric of the blood clot. So we add LPS and we see it binding to the clot. One piece of evidence. Second, next slide, please. And LPS also binds to the human blood clot. My first observations of LPS capture by extracellular blood clot was with horseshoe crabs. And then I tried it with blood clots formed from the blood plasma of my wife, who I showed you in that earlier slide. And you can see that it also beautifully decorates the fibers of the extracellular, in this case, fibrin blood clot. Next slide, please. And here is the formation of a human blood clot with purified human fibrin. And we also see that LPS decorates that clot, indicating that the binding is actually to the structural protein of the clot, fibrin, rather than to some other LPS binding protein that might also be decorating the fibers of the blood clot. Next slide, please. Here is quantification of binding of LPS to the blood clot. In this case, it's for the example of horseshoe crab, and the amount of LPS that can be captured by the blood clot formed from 1 milliliter of horseshoe crab plasma is, well, let's see, it's one bacterial cell contains this amount. Oh yeah, 1 nanogram of LPS will bond, it can capture the 1 milliliter of horseshoe crab plasma can capture the total LPS content of 17,000 bacterial cells. So it's a significant ability of the extracellular clot of horseshoe crabs to capture a substantial load of the toxin, endotoxin. Next slide, please. So how is the LPS binding achieved? LPS is a sticky molecule in the sense that it attaches to all sorts of inorganic or non-biological surfaces. However, LPS binds to proteins, to a very small subset of all the proteins that we have. And it binding is occasioned by capture of, by specific domains of the protein of close-spaced lysine and arginine amino acids, positively charged amino acids. When we block either lysine of the blood clot or arginine of the blood clot, we see the loss of LPS binding, suggesting, next slide, please. Here is an example of an LPS binding protein called FUHA, and it binds to, over here, lysine 306, lysine 351, arginine 382, and lysine 28 in close spacing that captures the phosphate groups of the diamino glycine that makes up the backbone of LPS. And when we, next slide, please. When we complex those, either the arginine amino acids, the amino groups of arginine amino acids with PGO, we see a loss of binding. This is the clot itself. And the next slide, please. Next slide, please. Oh, when we block lysine, we also see a loss of LPS binding. So to summarize then, LPS is a major cause of morbidity and mortality. LPS capture and inactivation is a major task of the immune system. And we have documented a novel, brand new mechanism for sequestration of LPS by the extracellular blood clot. The way we imagine this functioning is that bacteria, that the animal is wounded, a blood clot forms, the bacteria entrapped by the blood clot begin dying, releasing their LPS, but the LPS doesn't make it to the systemic circulation of the animal. It's sequestered there in the blood clot, where it's not going to cause the kind of mischief that disseminated LPS would wreak upon the animal. So one of the important mechanisms of immunity, one of the mechanisms that allows horseshoe crabs, lobsters, and people to survive in a highly septic environment, survive the challenges posed by bacteria. Thank you. That's it. And I'm not going to show all the rest of the slides. I promised I would finish on time.
Yes, Prof. Thanks for your presentation, Prof. I would like to open the floor is open after the second presenter, by the way, and the next presenter will be Professor Jerome from Chinese University of Hong Kong. He is currently as a professor at the CIHK Hong Kong, and we have been working together for a long time, and he's expert on genome biology. So please, Jerome, the floor is yours.
Right. Okay. Thank you. Thank you, everyone. So today, I'm going to talk about a slightly different approach. That I'm thinking about the conservation of horseshoe crabs in Asia. So if we're taking a look from the genomic aspect, so what would be the maybe we share a few ideas, for discussion. Or shall we do cross-regional genomic approach, or shall we do regional one, or how are we going to do? So this is something that I want to share my view with you in this talk. So first of all, of course, I would like to thank the organizers for gathering lots of other very good speakers. And of course, thanks very much for the invitation. So before I really start my talk, I'd like to thank my funders, my major collaborators on horseshoe crabs, and of course, I hope this list will keep on growing. So of course, Mark, Dr. John, Nova, Dr. John, Billy, Sita, Brian, Professor Portion, and also Professor Yuchi. And of course, I would also like to thank other organization partners like the AFCD, the IUCN, of course, the Ocean Park Hong Kong, and also WWF Hong Kong. So the question I want to address today is this. When we are trying to work on the conservation of horseshoe crabs in Asia, so shall we do it regionally or holistically in a cross-regional approach? I'm not going to express my views just because I think like this. What I want to go back is to be scientific driven. So I understand that this topic, whole genome duplication, we know horseshoe crabs have whole genome duplication, but I understand that they're all coming from very, very diverse backgrounds. So I want to step one step back to go back to the basic, and I hope you don't mind. For those who are very familiar with whole genome duplication, please bear with me. I want to do a talk on very, very basic to try to illustrate to you what is actually happening in horseshoe crabs. And because of that, the answers to the question that I have been addressing will be very, very straightforward. So if we go back to genetics 101, okay? So we know that, we are diploid animals. So we have all our somatic cells will be 2N, two copies, one coming from father, one coming from mother. Then during meiosis, when the sperm to get meat cells, sperm and eggs, then it will become N. And of course, sperm and egg meat will become 2N. I think that is very, very basic genetics we all know well. What are we actually mentioning when we talk about whole genome duplication? There are many different ways that can create whole genome duplication. I'm just going to use one example. I'm not saying that horseshoe crabs are under this specific mechanism, but I just try to illustrate the concept well. So imagine if this is an animal, for example, the diploid number is 2N equal to 6. So if it's going to do meiosis, it should go back to N equal to 3, right? Before the sperm and egg meets, and N will become 2N will become 6 again. But somehow, due to some errors, it could be meiotic errors, for example. And again, I'm saying that not horseshoe crabs under this. This is just one way to illustrate how whole genome could be duplicated. So N, in this case, will still be 6 instead of becoming 3. And the sperm and eggs, for example, they are all N equal to 6. You have an animal that will become N equals to, or N equals to 12, okay? So of course, to keep the very, very complicated story very, very simple, you imagine 2N equals to 2, when it goes to gamete cells, still 2N, that's fine. In your animal, you will have 4N, you duplicate your genetic materials. That is whole genome duplication. Your whole genetic materials get duplicated, okay? Why is that important? And this is actually so important, especially when you think about whole genome duplication in horseshoe crabs. One step back first. It has been long known. In invertebrates, the major differences people think about invertebrates and vertebrates is because vertebrates have gone through 2 rounds of whole genome duplication in their ancestors. What does that mean? So in the vertebrate ancestor, the genetic materials duplicate once and further duplicated once, okay? That means 2 times. So they have 4 times of genetic materials to invertebrates ancestor. That's what people have been thinking. And of course, I mean, like human ourselves, I mean, so that's why if you look into some single gene in invertebrates, human can go up to 4, for example. In fish, Helios fish, they have another round of genome replication. The situation becoming to change in recent years, to which, well, of course, majority of invertebrates do not have whole genome replication. There are few invertebrates that becoming to find their ancestor have whole genome replications. The two best examples that I'm going to illustrate today, one is the horseshoe crabs. I mean, in 2014, I mean, people sequenced the Limulus genome, found whole genome replication there. Of course, in 2017, people found that also in arachnid and spiders, there are also whole genome replication. And later, I mean, in 2016, we found that, well, in the 3 horseshoe crabs, CR, Limulus, and also TT, it's actually a whole genome replication shared in horseshoe crabs. What does that mean? In the ancestor of horseshoe crabs, genetic materials got doubled and doubled and doubled, okay? This is a very, very unique mechanism that we, using the diagram I shown on the bottom right, okay? Whole genome duplication is actually shared among the horseshoe crabs, also in arachnids, and also in vertebrates. And all the other animals, they do not have, okay? Very, very unique mechanism. And actually, because of this unique mechanism, when we consider the conservation of horseshoe crabs, we need to consider it slightly different, which I'm going to explain later. Recently, of course, another group in Singapore and together with our group, we showed that the horseshoe crabs, okay, the horseshoe crabs ancestor have actually gone through 3 rounds of whole genome duplication. Okay, go back to my previous slide. We only know they have whole genome duplication, but we don't know how many rounds. In vertebrates, the ancestor have 2 rounds, and the ancestor of horseshoe crabs, we have shown that there are 3 rounds. Okay, that means double, double, and double. That means 8 times of genetic materials, and to other, I mean, if you, it's very, very simple terms, than to other invertebrates. But that is not my major concern today. I want to show you how things actually happening in different populations. Allow me to go back into some basic genetics first. Of course, I will use very, very simple terms to explain this concept. Imagine this is just one gene, okay? One gene, it has 4 functions: blue, red, green, and orange, okay? Just imagine. If it gets duplicated, okay? So just imagine the gene get duplicated, it becomes 2 genes. So each gene originally can have 2 different, can have all carrying the same function: blue, red, green, and orange. But the fate, because during evolution, they go into different population, different individual, different population, the fate of the genes will be different. So some may go into subfunctionalization, which means each of the gene will get different function. Maybe this gene get red and orange function, the other one gets blue and green. So it's still similar to the original function, they just share the function. But it can also go into new functionalization, which is the case to which the original one keep the function, but the other one gain a new function, like brown and yellow. And of course, it can also be one gene remain, the other one being lost. The diagram on the right-hand side, I mean, coming from science, I mean, is demonstrating to you, for example, if genome get duplicated, you have gene function, they have many different gene function. If they have more function, they can go into many, many different ways, okay? Long story short, after gene get duplicated, of course, in the horseshoe crab, when genome get duplication, individual population can get different function for their genes. Now I'm going to illustrate with you the real examples actually coming from horseshoe crabs and how they actually happen. I want to first explain to you one type of genes, which is called the homeobox gene. Don't worry about the details. I mean, so the gene is very long, but the major functional part is 180 nucleotide base pair. They will encode for 60 amino acids. Okay, these 60 amino acids are highly conserved real animals. They work as transcription factors, which means they control many different genes. But the major is you can look into this 60 amino acids, which is called the homeodomain. This 60 amino acids is what I'm going to investigate. We have already published the paper in that communication biology, but I'll show you this 60 amino acid. We can use it as a clue to understand what has been happening in different populations. So case one. So of course, thanks to many collaborators, we have been taking horseshoe crabs coming from different populations in Asia. So this is the TT. This is the 60 amino acid that, if you look into the lower diagram, this is the 60 amino acid I've been talking about. So the one is here we are comparing the population in between Hong Kong and Malaysia. You take a look into this situation. We have a non-synonymous situation, which means if you take a look in the above area, the nucleotide change, but the amino acid do not change. Because the amino acid, if they change, it may change the control of the genes. So at nucleotide level, you see things are changing, but the amino acid do not change. But they're in different populations. This is the score. We take 20 individuals in Hong Kong. They are all like that. But in Malaysia, they have both types. You have some that gone into G to A substitution. You also have two. You really have some genetic diversity across Asia, but they do not change amino acid. Case two. I'm showing you another non-synonymous situation that is comparing in Hong Kong and Malaysia. This is also that you will change the amino acid from R to A, but they have a similar residue, so they do not change the function. You see genetic diversity actually happening. But what is more and very, very important is case 3 and case 4, which I want to illustrate to you. This is non-synonymous substitution, to which when you change the nucleotide, you also change the amino acid. And this is Hong Kong, Malaysia, Thailand, and India. So only in Thailand, you see this substitution has occurred in this homeobox gene. And majority in the Asia, like in Hong Kong, Malaysia, Thailand, and India, have the other form. And finally, another case 4. Of course, there are numerous cases. Just pick few cases. In this case, you have a huge deletion in the homeobox gene in the situation that we found in the situation in Hong Kong. So in the rest of Asia, of course, Hong Kong also have that. You have a functional gene because I've told you that these 60 amino acids are very, very important for controlling genes. But if you have a deletion, you have another copy. You lose the function. And this can become a pseudo gene. This gene is not functional anymore. In Hong Kong, you have among 10 individuals. This is CR, not TT. This is CR. So among the 10 individuals you find in Hong Kong, 9 of them actually lose. The gene becomes pseudo gene. You have one of them that is remaining like the rest of the age here. The point I want to raise, of course, there are numerous cases. First of all, the point we can actually see is that at genetic level, if you look into some genes, they are actually very, very diverse. We have something that are in common across Asia in different populations. You also see unique, for example, in Malaysia, in Thailand, or in Hong Kong. This is actually a collaborative study with many colleagues in Asia. So we get the individuals from India, Thailand, Malaysia, Hong Kong, etc. And if we just use the normal population genomics, just taking a look, you also see every population is very, very distinct, okay? So I do a quick summary first before I go back into my question to see how we can move forward. Horseshoe crabs shared an ancestral whole genome duplication. Unlike other individuals, that is very, very special and unique. We have proven, together with other colleagues in Singapore, that 3 rounds of whole genome duplication have been occurred in the Asia horseshoe crabs CR and TT. Distinct populations are reviewed in different places in Asia. Of course, also common together, not only just distinct, we also share something very, very common. And at the genetic level, there are various dynamics and evolution of genes in different populations. Even if in small places in Hong Kong, different places in Hong Kong, you see different population as well. So, I missed one slide. Oh, that is the older version. Sorry, let me see if I have them. I have this slide, okay. So, what do we need? I go to this slide first. I think it is very, very important to understand the genomics to inform us what actions to be carried out. This is very, very important if you understand what I just tried to explain. First of all, to consider what are the areas to be protected, we also need to think about what are the distinct populations to be protected. This we need to be backed up by scientific-driven policy. So there should be understanding the genomics. Also, when we think about breeding population, so we know many groups try to breed their own population, but when we are trying to breed that population, what is that population representing? Is it representing the dominant one in the field or distinct population in the field? This is something that we need to come back into science to understand the basic. And of course, especially when we try to release them back into the environment, shall we be releasing something that is genetic very, very distinct to increase the genetic gene pool? Or maybe we should be releasing something that should be genetic more similar. That will be depending on the situation regionally. And of course, also globally. We need to think about them in a very holistic approach, in a collaborative manner. Otherwise, we may be doing, if we do it the wrong way, we may be doing something not so good. So not only in particular area regionally, we also need to do cross-regional because there will be overlapping areas. For horseshoe crabs and in Asia, of course, for example. So we need to manage, how do we manage overlapping areas, especially those populations, what does it representing? As I have already mentioned before, introduction of genetic diversity, or we should be releasing something back into the environment, something more similar. And of course, shall we be keeping one breeding line, numerous breeding lines, and how about trading of breeding lines? This is something very, very forward-looking. I hope in the 5 to 10 years, we need to be doing that very, very systematically, such that we'll be doing in each area. We are doing, for example, Hong Kong, then we do Hong Kong, of course, but we also need to collaborate together with many people in Asia, trading, etc., exchange of information. The question is how? I think we need to work together as a community, which I'm sure we are working as already, thanks to Mark and Paul. The IUCN network is good. But we need to recognize, respect, sharing of expertise, data, and samples in a transparent way. Nobody knows everything. If you ask me to breed horseshoe crabs, I need expert, okay? If you ask me genomics, yeah, I'm very happy to share. I'm very happy to do it. The community can start with academics, but not only academics, government, NGO, public sector. How can we do? This is something that I've been thinking, which is now, thanks to the Research Grant Council, so myself and together with Dr. Zhang and also many colleagues in Hong Kong, we have acquired a sequencer, a genome sequencer that can be in Hong Kong. So it will help us to understand the genomes of different populations in the wild in Asia, and also the breeding population organized within individual labs. More importantly, we need to construct a sharing platform to which not only us know, but we will be able to train you to understand how all this population genetics or the line that you are breeding. So we need to get that, and then will be another higher level of policy and say, oh, maybe in Malaysia or the genetic diversity is not that much, for example, or Hong Kong they are much, or the other way around. If we change, or maybe what we should do, we need to form a peer network to govern how are we going to use all this information. And from that, using scientific-driven genomics will actually inform us how to do the rearrange breeding programs and release program. Of course, working hands in hands together with NGO, etc. And of course, I mean, my lab, I mean, we have also other proteomics, gut microbiota, I mean, and also eDNA, and also citizen science. I think this platform can include all of them, really sharing all the information such that the community can grow stronger and also exchange in a very, very open way. So back to my original question that I want to address in this talk. When we are working on conservation of horseshoe crabs in Asia, shall we do regionally or holistically, cross-regionally? The answer is, if you understand, I've been talking about the genomics, about the different individual, different populations, they have distinct but also share. So that means we need to do both in a collaborative way. So again, I mean, I thank the organizers for giving me the chance to share my view with you. And yeah, thank you very much. That's all.
Thank you, Jerome. I would like to open the floor for the question for Professor Peter as well as Jerome.
Let me stop sharing first.
Yeah. If there is any question, please proceed with your question, unmute yourself and ask. Professor Barton. Yes, Prof.
Hi, I was interested to ask Professor Armstrong a question. The systems that you described are only effective against gram-negative bacteria. So what happens in the case of gram-positive bacteria?
Prof. I'm looking for Professor Peter is not online, it seems. I'm sorry, maybe is some technical issues, perhaps, Prof. He's not in the discussion. Is there any other question? Sorry. I have a question to Jerome regarding the whole genome duplication steps. How about the samples itself? For example, if we have a different number of samples which are collected from different locations, will it interfere with the, I mean, will it give the different data on the whole genome sequencing or whole genome duplications? I mean,
yeah, I think, I mean, the beauty of horseshoe crab itself, when we think about the biology part, it's very, very interesting because it's very, very special. But how is the situation for whole genome duplication happening in horseshoe crabs? Is it something unique or mirroring what has been happening in the vertebrates? I think this is completely an underexplored situation. And of course, also of course, I understand people in Singapore has already been doing in Tang here as well. But population-wise, what is happening? I think this is something underexplored. And so the baseline information is also not well known. I think we should move into that direction. Population genomics, as well as understanding whole genome duplication effects on horseshoe crabs. Of course, not only in Asia, in America and Limitless, the situation is also unknown. It's very, very pretty that horseshoe crab has such that can contribute much knowledge to the biology, not only to horseshoe crab, but overall to textbook, if you like.
thank you, Jerome. Is there any other question? One more question is welcome. I think we can move on to the fifth presenter because of the time constraint. We have 1 more hour to go. The next presenter will be Professor Chong. He is joining us from City University of Hong Kong. He is a full professor in the Department of Chemistry, and he's a member of State Key Laboratory of Marine Pollution, and he is also the steering committee and the founding member of IUCN SSC on horseshoe crab. The floor is yours, bro.
Okay, can you hear me clearly?
Perfect.
Okay, good. Thank you, Dr. John. So first of all, I would like to thank the organizing committee for providing me an opportunity to share with you some of the community outreach programs in Hong Kong. So today I'm going to share with you the activities organized by different NGOs and the lessons they have learned and the difficulties they have encountered. So this will give the colleagues from other countries and regions some ideas what Hong Kong is doing now, and whether anything you can learn right from this kind of activities. So first of all, right, let me talk a program that some of you may have heard about, which is the Horseshoe Crab Rearing Program, organized by the Ocean Park Conservation Foundation in collaboration with the City University of Hong Kong. As mentioned earlier by Dr. Paul Shin, this program started in year 2009. So it has been running for 12 years. And this is also the first public outreach program organized in Hong Kong, particularly for the horseshoe crabs. So what this so far, right, up to now, in fact, more than 150 schools have participated in this program with over 6,300 students. And these students, they not only study horseshoe crabs in the laboratory, they also deliver the conservation message to their communities through, for example, a school open days. And in fact, because of these community outreach, so they have reached more than 206,000 people, right, in their communities. So I would say this is quite a very successful program, both in terms of the students participated and also the people they have reached. So what this program is about, I'll give you a brief idea. So my university got some laboratory bred horseshoe crabs, and then we delivered to the schools, and also we have a research assistant supervising their work. So the students have to keep the animals in the laboratory for about half a year. And during that time, they have to monitor the growth of the animals as well as maintaining the water quality of the supporting system. So at the same time, here is a photo showing you the student actually was measuring the salinity of the water, and another student on the right here actually is feeding the horseshoe crabs using the brine streams. And so after about half a year, so hopefully, right, if the crabs they grow well, and so different students from different schools, they gathered together and went to Har Park Night to release the juveniles in a summer event. But before that, the OPCF also invited students to do a cleanup activity on the shore. Because later on, I'll talk about the problems occurring on the shore. So they have to clean up the beaches and as a kind of environmental education. And then, okay, after cleaning up the beach, then the students will release their juveniles as shown here, right? The tiny ones, they have been keeping for half a year. So very often when we do this kind of education program, of course, you may ask, okay, we have done something, but whether this kind of work is effective or not, and what are the goals these programs have achieved. So in order to systematically understand the the impacts of this study. So we review the data, particularly by the work from Zhou Cheung and Eric Collor at the time from the Ocean Park Conservation Foundation. They actually did surveys with all the students, with students participated in the program before and after, right? The program to compare their knowledge and attitudes, behavior, etc. And this was published in this paper. So of course, if you are interested in get a copy, you may email me. So we analyzed the data covering the period between year 2009 and year 2016. And basically, there are three main changes observed among students. The first one is their knowledge on their biology and ecology was substantially strengthened. So that means they have better idea what horseshoe crabs are and where do they live, what do they eat, okay? At the same time, there's also changes in their attitudes and perceptions. For example, they become more responsible because they have to take care of the animals to make sure that they grow healthy and happily. And at the same time, there's also changes in their behavior. For example, their self-confidence was enhanced because they had the chance to give public talks, to introduce the conservation message to the general public. But still, there's some changes among these aspects. And the most significant change was the ecological knowledge. That means the study found that the student has substantially increased their understanding of the animals, and then followed by changes in their perceptions and attitudes. And the changes of behavior, although there's some changes, but it's not as much as the knowledge they gain. This is natural because very often, if you want to change the behavior, very often you have to know, right, something, and then you will change the attitude and then affect their behavior. That's very normal, right? As shown by psychological studies. So based on that, of course, we know that this program is effective in changing the attitude of the students. They become more environmental aware, and they also to be more committed to conservation. So of course, the next step should be how we can strengthen the program. The first one, of course, is to provide more opportunities for students in engaging other local conservation initiatives. For example, doing population survey, participating in workshops and exhibitions. And at the same time, of course, it's good, right, to incorporate the conservation of the horseshoe crab into the school curriculum. Especially nowadays, we are talking about STEM education, right? The science, technology, engineering, mathematics, right, which are important for future community development. So in fact, horseshoe crab can be a good case study, right, in this kind of STEM education. And this is this program actually also I've got a Facebook page and Instagram, right, to deliver a message not just to the students but also to the public. So this can also arouse the public's awareness and understanding of this animal, right, through these media resources. And the education program actually extended to primary schools organized by the Ocean Park Conservation Foundation. This was the program done last year. Of course, there's some problem because of the COVID-19, but they still have some school visitors. And in the schools, they organize this kind of online communications at the same time through this kind of platform. Okay, so this is the staff from the OPCF, and then they communicate with the students through this online platform because of the COVID-19. But they also have the students also have the chance to get hands-on experience and knowledge of these animals, and they also have different play stations, right? So they walk around and to learn different kinds of knowledge on the horseshoe crabs. So this is a quite more interactive way in delivering the message to the primary school students. But there's some problems as far as we know. First of all, because of suspension of the school due to COVID-19, And many of these activities actually have been suspended, including our field trip, public seminar, and wild release. And some of this workshop and training have to be done online. So that inevitably this will affect the interaction between the speakers and the audience. And on the other hand, particularly in the primary school, because there are games associated with some education objectives, they have to set some stations, but the problem is different schools, they have different settings. So that means the communication before, right, the actual games are held, right, should be improved, because there's maybe some kind of misunderstanding between the staff and the teachers. So that means this has to be considered in the future when we try to organize this kind of interactive games. And another activity organized by the OPCF is a regular population survey. In fact, as mentioned by Dr. Paul Shin, recently there is an observation network organized by Billy from Guangxi. But in fact, the OPCF has been doing a regular population survey at several sites in Hong Kong where highest density of horseshoe crabs are found. So they recruit secondary and students as also as students from universities to do survey every 3 months. Basically, it's in March, June, September, and December at several sites. But the problem is when organizing this kind of activity that involves students from different schools, secondary schools and universities, it's very difficult to find a common time schedule, right? Common time slot for all the students, particularly in the training, and that create problems. So the staff, the OPCF, staff from the OPCF actually has to take extra effort in organizing the training for different students group, because these students they are available at different time of the in the period. So these are some kind of coordination is required, and extra effort is also required. And also, of course, how to do the survey, right? It's a problem. You have to let the students know clearly and correctly and scientifically how to do a survey. And but the problem is students, they come and go. So OPSF actually suggests later on that it's better to identify a pool of helpers so that they get familiar with the survey techniques, and then they can act as a big brother, big sister to introduce the skills, right? They can pass on the skills to next group of surveyors to save time and the effort of the OPCF staff. And of course, in Hong Kong, weather is also a problem, particularly during summertime, which is extremely hot and humid. It's very difficult to do in the field work. So that may reduce the number of survey days, right, for this study. And then I want to introduce you another project. This time it's not much related to education. It's more related to volunteering work and management work. Actually, is the work by the Nature Conservancy, the TNC, particularly at this site we call the Pet Night. So this study, in fact, before we talk about their work, here is a map that we did for the government in population survey in year 2018 and 19. In fact, right, we have similar results obtained in terms of the distribution of horseshoe crabs in Hong Kong. Red one showing the Chinese horseshoe crab or transparent horseshoe crab, the blue one representing the mangrove horseshoe crab. Here actually in we call the Ha Park Night and Park Night has the highest density of horseshoe crab, and this is actually what we are going to talk about. The work done by the TNC. In this area, there are several problems. First of all, you can see this is the mud flat, and with the mangroves where you can find very high biodiversity, but they are not protected, right? They are not protected by law. And on the other hand, there's unregulated tourism, so that people come and go, and many of them, if they visit the mud flat and they go there and enjoy the sunshine and digging up the animals, etc., all kinds of activities, but the activities are not regulated. And also you can see a large amount of this marine debris and also aquaculture debris, including oyster shells. And also there's some invasive grass species that cause problem because the grass they grow very rapidly and will degrade the habitat. So because of this, at the same time, this is an area where high density of horseshoe crabs are found. So one more thing is about the oyster shell. I want to speak more. In fact, this area we call it Deep Bay, where you find the Parklight is the only area where oyster culture is found. And this is a typical oyster culture farm on the mudflat in Hong Kong. So people, they put a concrete post there and then attract the larvae, swimming larvae of the oyster to settle on the post. And when the animal reaches certain size, they will transplant these oyster to the floating rafts. But on the other hand, because of these aquaculture activities, it's left behind a large amount of these oyster rubbles, as well as the abandoned, what we call the oyster cultures like this one, right? So these are the abandoned posts, the concrete posts for the settlement of the oyster larvae. So because of that, we did a study at the City University of Hong Kong to try to understand whether there's any problem created by these kind of concrete posts and oyster cultures. So we use bricks to simulate these oyster concrete posts. And then after 3 months, we find there's some changes in the sedimentation, seeing there's some settlement of the sediment between the blocks. And this will affect the movement of the horseshoe crabs. And in fact, we record here comparing the area with bricks and the area without bricks, which is the control. You'll see the density of juveniles actually decrease. So that means actually, because the horseshoe crab, they don't like the area which is dry. So during low tide, they only stay in the water pools. So because of the change in the landscape, in fact, because in the juveniles, they tend to move away from this area. So that means oyster rubbles and poles actually will deter the juveniles from settling in that area. So because of that, actually the TNC has initiated a program to improve the environmental quality. First of all is what they propose is to do an oyster farm reconfiguration. So they remove some of the oyster shells and the abandoned posts. So they pile up together, right, and turn it into a kind of reef structure to attract the biodiversity. So because other animals, they may live there as a refuge, or they settle there because of the increase in the food amount. And they also help to remove these grass species, the invasive grass, as well as to take away the posts, all these activities. On the other hand, they also include the academics, for example, professors from the Hong Kong University, so to do some kind of horseshoe crab population survey and to see how this reef structure changes at sedimentation and the biodiversity. So that involve both the volunteering work in removing the debris and the abandoned poles, clean up the beaches, but at the same time doing some ecological monitoring. This is the one that they talk about, is about the abandoned farm reconfiguration, right? This kind of reconfiguration does here is the abandoned oyster farm with all these poles, right, lying on the mudflat, just like the photo on the right. So they try to change it by removing these poles they pile them up into different areas, just like this one, right? They pile them up and to act as a kind of you can say artificial reef, right? To create a reef that attract animals staying there, and also may change the sedimentation and hopefully can increase the food supply. And then they remain some areas, right, as a kind of a demonstration, right, of the oyster farming. So these kind of activities are ongoing. They just started the work a few months ago. So we are still waiting for the results. But on the other hand, they also involve some local communities in this, and also organize tourist eco tours, right, to spread the message of this kind of oyster farm reconfiguration. And then, so these are the points they consider valuable. First of all, it's important to engage stakeholders in the early stage. I mean, before they change any settings, right, on the abandoned farm, they have to engage different stakeholders. For example, the oyster farmers, the villagers, and the other NGOs, or even the scientific expertise. And of course, there are some problems that when I deal with multiple stakeholders, because they have different agendas and they have different priorities. But anyway, of course, this is a good experience in initiating this kind of work. And then another one I want to talk about is the one organized, just finished, right? Almost finished, I would say. It's a program organized by the Worldwide Fund for Nature Hong Kong, which is called the Horseshoe Crab Friendly Campaign. So far, what we are talking about, like involving schools and involving volunteers or improving the habitat quality, all are talking about the juveniles, horseshoe crabs. And this one actually focused particularly on the adult horseshoe crabs. Now, in Hong Kong, we have no idea where those adult horseshoe crabs are. They live in the sea, but no one knows where they are, how many they are. So this campaign, of course, try to fill a gap, right, with regard to the horseshoe crab. First of all, they did several surveys. The first one is a survey done by an independent institute. So that helped with the survey. So they telephone interview more than 1,000 citizens about whether they know what horseshoe crabs are. Have they seen any horseshoe crabs in Hong Kong? And so these are the major findings. First of all, it's interesting, right? Even though we have been running the program for 10 years, more than 10 years, half of the citizens in Hong Kong, actually, they have never heard of horseshoe crabs before. They know nothing about horseshoe crabs, what horseshoe crabs are. But anyway, if we told them, when we told them what horseshoe crabs are and show them the photos, in fact, most of the people, 99% of them, would not consume this animal for any purpose. Disagree with the questionnaire Dr. Shin and I did, right, in year 2005, that showing that, in fact, consumption, right, is not a problem for horseshoe crabs in Hong Kong. In fact, people in Hong Kong, they don't consume horseshoe crabs. And then the last result of this survey is 52% of the public were more willing to choose seafood restaurants that opt for ocean-friendly practice. That means, in fact, at least half of the citizens in Hong Kong, they are aware of the problem, and in fact, they're willing to choose horseshoe crab-friendly restaurants or seafood-friendly restaurants. Okay, so based on, in fact, at the same time, right, in addition to knowing the perception of the general public, they also did a market survey and tried to see whether they could find any adult horseshoe crabs in the seafood restaurants and the stalls. So they visited 98 seafood restaurants between October 2020 and March 2021. They just finished the survey. And in fact, very few restaurants and stores, right, they display horseshoe crabs. Among those studied, there are only 7 of the stores actually with horseshoe crabs, adult horseshoe crabs. But interestingly, just half of them were transparent horseshoe crabs. So that means the other half should be the mangrove horseshoe crab, because we have only 2 horseshoe crab species here. But of course, we know, right, the mangrove horseshoe crabs are toxic. So if they display in those stores, I won't imagine, right, human actually would take these animals for consumption. But maybe these fishermen, they harvest the animal by accident, and then they just display, they sold to the restaurants or stores for just display. And then another one is they also try to get some history of the horseshoe crabs in Hong Kong by interviewing the fishermen. And the fishermen actually being interviewed, telling them that the horseshoe crabs are non-target species. That means the fishermen actually they won't harvest horseshoe crab for purpose. And so only they are harvested only accidentally when they were doing trawling. But on the other hand, the fishermen also agree that distribution and number of other horseshoe crabs are decreasing. So that means the decrease is pretty obvious, and they can find the difference. And on the other hand, in fact, the horseshoe crab sales, right, are not high. They couldn't get any major profit. So that means these animals actually are not attractive to the fishermen. But anyway, the fishermen are also willing to release the bycatch back to the ocean. So that means that it seems that the fishermen, they're also doing a good job, right, in conserving these animals. First of all, there's no market in Hong Kong. They can't sell it at high price, but on the other hand, they're willing to release these animals. And the last one is interesting is, in addition to doing a survey, the Worldwide Fund also encourage the seafood businesses to pledge not to sell or display horseshoe crabs. And so they design this kind of labels and help to post them in some areas as a kind of pledge. And in fact, they have convinced 26 stores to join the pledge and put the stickers on their walls. And in fact, this is a good starting point. But there are some challenges. The first one is because now we are still being affected by COVID, that now the condition is getting better. But a few months ago, in fact, the restaurants were not allowed to open at nighttime. And that's, of course, a problem because that will affect the seafood stock pattern. Some kind of seafood, they will stay in the restaurant for a long time because no people go there and have dinner. And also because of the economic downturn because of COVID-19, so many shop owners, they were worried about their income, right? Because not many customers visiting the restaurants and buying seafood from their stalls. So in fact, they were not interested in this kind of educational message because they're afraid that may affect their income. But anyway, at the end, right, in fact, there's an interesting finding. They can convince at least some of these stores, right, to join the pledge. Basically, it's because they work with these stores and explaining that the campaign, in fact, can be a free promotion. Because based on the survey of general citizens, many citizens, in fact, are willing to visit those restaurants that are more aware, right, of the conservation issues. So that means it helps in branding, right, those restaurants. So it can be a free promotion. So because of that, some restaurants have agreed to join the pledge and allows them to put the sticker on to notify people that this restaurant, they won't sell any horseshoe crabs. So this is a very good move. So the last one is my own work, just finished about a year ago. So this is a study that involves citizen science in doing surveys and on horseshoe crabs and marine litter on the beaches. So in this, this is the site I work on. Is in Shui Hao, which is on Lantau Island. Is the largest piece of sand flat in Hong Kong with horseshoe crabs. And so we recruit some volunteers from both their general publics or students. And then we organize seminars, talks, and also field trips. So teaching them how to do a survey just like this one. So they have to walk along the transit line and then use their GPS to fix the location of every horseshoe crab they found, right, on the mudflat along these 4 transit lines. So this is how we in the seminar, we introduce the method of this population survey and then taught them how to identify the feeding trails of the horseshoe crabs and how the feeding trails of the horseshoe crab are different from a trail made by a gastropod. So this kind of knowledge, right? And then, of course, we also tag the horseshoe crabs because we would like to know, okay, because we went there for 30 times during summer. We visit them the shore regularly. So hopefully we could identify individual horseshoe crabs and eventually to work out the home range, right, of every individual. So we teach them how to tag the animals, and we teach the public how to use the meters for measuring temperature, pH, and also salinity. So all workshops and field trips are organized, just like this one. A field trip, introducing the meters for measuring water quality. At the same time, here we also took them to the mud flat to show here is a feeding trail, right, of the horseshoe crab, to get a feel of what horseshoe crabs are doing. And this is, in fact, this is the one actually they are doing the work in the field. You see, this is the feeding trail, right, of the horseshoe crabs on Shui Hao. And then this is the actual work they were doing in measuring the carapace width of this animal. And then another one actually was recording the data, okay, showing the actual work. But on the other hand, this is also, the site is also polluted by the marine debris. So there's also special trips where the people visit the sites for collecting marine debris and cleaning up the beaches. But more than that, we also quantify different types of marine debris, whether they belong to food containers, plastic bags, or the floats, the fishing lines, etc. So this is to quantify the marine debris, the types and the quantity. So this is the work we've done. In fact, so this is one of the photos. We collect the marine debris from the shore. But there's one, this is good, right? Because the people, they know the general ecology of the crabs, how to sample them, record the data, measure water quality, collect the debris, and quantify the type and composition of the debris. But there's one major problem in the population survey that I want to share with you, is because they don't have much chance to observe this animal in the field. First of all, because not many beaches in Hong Kong have horseshoe crabs. But on the other hand, because they know this is a survey, so they have an intention is to discover as many horseshoe crabs as possible. So that animals, sometimes they are so passionate in looking for horseshoe crab, in searching for horseshoe crab, that they actually deviate from the actual survey area, right? That means they didn't follow along the transit line. They didn't follow the guideline in doing the survey, because they try to recover as many horseshoe crabs as possible. And sometimes we couldn't, right, keep asking them to follow the guidelines because they were so enthusiastic about this kind of survey. But anyway, of course, this is a good experience, but to them, but from the citizen science point of view, right, the data actually have to be interpreted with caution. So, and on the other hand, they also study the human impacts on that shore, because during windy time, people, they do this kind of windsurfing in this area. We have no idea whether this kind of activity actually affect horseshoe crabs, but at least, right, they have to record how many people visited there, doing what kind of activities. So to try to see whether there's any potential conflict between these human activities and horseshoe crabs. So here, just give you a brief overview of all the horseshoe crab work being carried out by different NGOs in Hong Kong. So I want to end my talk with the quote from Howard Zinn, a philosopher from the United States. So I think every one of us can do a little bit, and hopefully this can transform the world. So thank you very much. So I would like to see if you have any questions later on after Kevin has done his sharing. Thank you very much.
Thank you, Prof, for your presentation. It's enlightening. I would like to move on to the final presenter of this session, Mr. Kevin Lowry. He is the person in charge for updating the red list assessment for all the Asian horseshoe crab, all the three. For tri-spine horseshoe crab, the work is done. For the other two horseshoe crab species, we are still working on it, and he will be sharing some insights on CR Central. Kevin, the floor is yours.
Can you hear me?
Yes.
Okay, great. Okay, first of all, I have to apologize. I'm not a professor, so I'm not going to be doing a slide presentation. And that's also because I'm sort of technically challenged. And as a consequence, I dare not try and do a slide presentation at the same time as I'm doing the my own presentation. So I'm going to cover the CR Red List assessment. This is a review of where we are and what we've done to this stage. And the presentation is going to cover the geographic range, biology, habitat and ecology, populations. I'm going to talk about the three major threats that we've identified so far, and then I'll summarize the key issues. It has a west to east range from the coastline of Odisha state on the northeast coast of India, all the way through much of Southeast Asia, the Sundarbans, Bangladesh, Myanmar, Thailand, Malaysia, Singapore, Cambodia, through Vietnam, China, Hong Kong, Brunei, Dar es Salaam to Indonesia in the east. And its range is largely coincident with the west to east range of T. gigas, and it overlaps in some places with the range of Tridentatus, the tri-spined horseshoe crab, which extends from north to south. In comparison with its peers, whilst its biology and ecology have been well studied, the populations are understudied for a number of reasons. Mangroves are pretty inhospitable places, and mangrove mudflats can be difficult and sometimes dangerous places to navigate. Mangroves also in some areas of the range are homes to dangerous animals. In the Sundarbans, for instance, the species range overlaps with that of the saltwater crocodile and the Indian tiger, and surveys have been done there which have avoided the range of those species. Surveys in Singapore have also been halted because of crocodile sightings. It's also a secretive species. It's not easy to study. It's not like the American horseshoe crab, which comes out on the beach and waits for you to tag it. You've really got to look for the adults. And finally, it's not economically important. So it hasn't been the focus of study grant funding in Southeast or in Asia. And that's quite important. You know, if you haven't got money, you can't study these things. In terms of biology and ecology, all four horseshoe crab species generally follow the same life history protocol related to spawning, larval development, juvenile foraging and development, subadult life, and then adult life. All recent horseshoe crabs, including Coscinoscorpius, are limited to a relatively narrow coastal area, and their distribution is discontinuous. And whilst the other three species favor what I would call oceanic conditions, its lifestyle is linked to estuaries and enclosed bays, and as its name implies, it's associated with mangroves. But it's not universally distributed in mangroves. For reasons as yet unknown, it appears to be site selective. In a 1989 to 92 study of 40 randomly selected what looked like promising sites in the West Bengal Sundarbans, it was only recorded at 14 sites and only in significant numbers at 3 sites. It's not clear why this is, but it might be related to an affinity to certain species of mangroves. And then in Singapore, studies have indicated that actually inside mangroves, it has microhabitat preferences as well. So even where it is in a mangrove, it doesn't cover the whole mangrove area. Spawning events take place on the high tides of full and new moons. The eggs are deposited in clutches in the muddy banks of mangrove streams or along the fringes of mangroves close to the high tide mark. And whilst the adults and juveniles can endure quite a high range of salinities, including fresh water, it appears the eggs cannot. They develop in a fairly narrow saline range. And this means the species is reliant for spawning on an incredibly narrow interface between sea and land. The first phases of larval development take place inside the egg, and once the larvae hatch, they develop further in the nest area before being carried to intertidal juvenile foraging grounds on ebbing high tides. The juveniles then forage for up to 10 years, maybe more, in the intertidal areas of mangrove streams and mudflats surrounding mangrove stands, migrating seawards as they get larger. The subadults inhabit the subtidal zones below the mangroves, and once mature, the adults move into deeper waters, returning to shore as amplex pairs to spawn. Migration is limited in adults. A tracking survey in Singapore in 2012 revealed that 11 out of 12 CRs only traveled up to about 6 kilometers from their original point of release. In terms of population genetics, where the studies have been conducted, there is evidence for genetically distinct subpopulations across its range. In Indonesia, high levels of genetic differentiation among local populations. There are 4 distinct populations. Populations from North Sumatra, East Sumatra, and Kalimantan do not share any haplotypes with each other. And then there's low levels of genetic differentiation between the populations in West and East Java. 3 well-defined populations have been identified in the Sundarbans, and there's a clear and large genetic break across the Thai Malay Peninsula. Obviously, it's not the peninsula, but something underwater is causing that genetic break. The marked genetic diversity in CR may be due to its affinity for its estuarine habitats. It's obviously less inclined to enter open oceanic waters. And this estuarine bound ecology has resulted in restricted gene flow between populations. These findings, or the genetic surveys, suggest CR populations are locally adapted. They're indicative of low levels of migration. And because it is a shallow water species, such traits will ultimately lead to population fragmentations. In terms of population surveys, the first surveys of the range and abundance of Carcinoscorpius were conducted in the mid-1970s to mid-1980s. Baseline population data does not exist across a lot of its range. There's no baseline data for Bangladesh, Brunei, Dar es Salaam, Cambodia, Thailand, or Vietnam. There are, however, good data from the rest of its range. In terms of national assessments, it has been assessed as being vulnerable in Bangladesh, Singapore, and China. It's currently being assessed in Hong Kong, and I'm still awaiting a copy of the Indian assessment, which has been done but is very difficult to find. In terms of adult population counts, quite interesting information is available. In India, the populations are well studied, and obviously you've got the northeast coast of India. The coastal area is on the periphery of its range extension, and then in the Sundarbans, yearly surveys were conducted in the mid-1980s. A survey in 1985 to 86 recorded 1,700 individuals, but the counts ranged from between 4 to 350 individuals at specific locations. So not very large populations at specific locations. And these small population counts in the Indian Sundarbans have been consistent over time. It's also important to remember this wasn't a daily survey, this was a yearly survey, a year-long survey. So not large populations in the Sundarbans. There's no baseline data available from Bangladesh, but scientists that I spoke to who do other marine biology estimate the population declines of between 40 to 50% in the last 2 decades. The first baseline survey was conducted in Myanmar between January to April in 2019. And after interviews with fishermen, 5 potentially good sites along the coast were surveyed. They found 10 male and 4 female CRs across the entire nation. That's 14 individuals for the entire country in that 4-month survey. Apart from adult surveys in China, Hong Kong, and Singapore, juvenile population counts and density studies are also conducted. But in some cases, these surveys have revealed the presence of small, sometimes statistically insignificant juvenile populations. There's not enough juveniles to actually do statistics on. For example, in 2009, only two juvenile foraging ground sites in Singapore had population sizable enough for population density studies. In Malaysia and Indonesia, again, the adult populations where they've been counted, they're small, from the low tens to the low hundreds. A 2016 survey across the northern coast of Java, and Java's the most human populated island in the world, found only 170 individuals. Between 9 to 50 individuals were recorded at specific surveyed sites. So, you know, we've got very small, very discrete populations across its range. In terms of threats, I'd like to talk about 3 significant threats, and these will relate to the biology and ecology, which I've just talked about. The threats from fisheries, aquaculture leading to mangrove loss, and climate change. In terms of fisheries, CR is not a targeted species, but it does constitute a significant component of bycatch for the following reasons. Spawning takes place on the high tides of full and new moons. Fishermen across the region set their clocks and deploy their nets according to the lunar cycles. In Hong Kong, I've spoken to fishermen who can tell you to within the hour when they're going to set their nets based on the full and new moons. You know, it's a clock that they just automatically react to. CR migrates up estuary and river mouths on new and full moons to spawn. The fishermen deploy their nets in estuary and river mouths on these incoming full moon tides. The adults traveling during spawning season, the adults navigate the intertidal and inshore areas. Fishermen deploy their nets across intertidal and subtidal areas on high tides. And whether it's India or Hong Kong, they often deploy nets which are hundreds of meters long to catch all of the animals that come inshore on the receding tides. The nets basically act as a wall to prevent anything from leaving. In inshore waters, fishermen deploy their nets in inshore coastal waters. Subadults and adults forage in the subtidal and inshore areas. A particular concern is the blue swimming crab fishery. The blue swimming crab range is coincident with that of CR. This is a gillnet or trammel net fishery, which is conducted in inshore coastal waters, and surveys conducted in Thailand and Indonesia have revealed that when conducted in the vicinity of mangroves, CR bycatch can be very high. Sorry. And finally, in offshore waters, throughout most of Southeast Asia and South Asia, trawlers fish offshore, and outside the spawning season, the adults forage in the deeper waters offshore. So in summary, all of the adult habitat areas lie in targeted fisheries areas. The second and probably single biggest threat to CR populations actually comes from aquaculture, and in particular that of the black tiger shrimp or black tiger prawn, Penaeus monodon. Penaeus monodon is economically very valuable to coastal communities, but its production is reliant on what's called the trap and hold method, in which a dike is built around a section of mangrove estuary, creating a pond. As at high tide, a water gate in the dike is opened and water carrying larval shrimp flow in to fill the pond. The gate is then closed, trapping the water and the juveniles inside. They grow, and several months later, the gates are opened and the mature shrimp are caught in nets as the water flows out of the pond. These trap and hold ponds are through necessity located on the spawning habitat and juvenile foraging grounds of CR. There's also an extensive industry of fishing for larval shrimp on high tides to supply these ponds, and that leads in places like in India and Bangladesh in particular to considerable amounts of discarded bycatch, which will include larval CR, juvenile CR, and adult CR. So one of the consequences of the sighting of these ponds is mangrove loss. And obviously the mangroves are critical spawning and juvenile development habitats. If you lose these, then you lose the entire capability of the species to reproduce. In Bangladesh, in the Chakaria Sundarban, there was an 87% loss of mangroves between 1972 and 2017. It's almost a mangrove desert now. In Myanmar, a 56% loss between 1950 and 2011. In Cambodia, a 10% loss between 2000 and 2010. Vietnam, 68% loss between 1943 and 2012. And 56 of the remaining mangroves in Vietnam are considered as planted and to be of low ecological value. China, 66% loss between 2008 and 2012. In Indonesia in 2007, 27% of the mangroves were in good condition, 48% in poor condition, and 23% were damaged. Now, the thing about this is, these are all government figures. I've taken all of these from the NBSAPs of the various countries. So these are actually conservative figures for mangrove loss in these countries. The major threats to mangroves obviously include reclamation, conversion into settlements, building of roads, ports, and other infrastructure development, illegal logging. But one of the main causes of all of this mangrove loss and degradation is aquaculture, and in particular, Peneus monodon aquaculture. The final serious threat is climate change. And two aspects of climate change pose serious threats to CR. Everyone thinks about the impacts, and I'll talk about those, but more importantly, what people miss is the government responses. So if you read the NBSAPs, all of South and Southeast Asia every single country considers themselves vulnerable to climate change. And this is manifesting itself in a number of ways. First of all, you have floods and droughts inland, storm surges and coastal flooding, sea level rise, coastal erosion, and then something which is increasingly being highlighted is saline incursion. So the government responses to this already throughout most of South and Southeast Asia, you see the building of dams, thermal power stations, hydroelectric power stations, and irrigation projects. And these are already affecting the downstream flow of water, of sediments, and of nutrients. And in fact, Mishra alerted to me to the significance of saline incursion when he explained to me the problems being caused by the Farakka barrage, which had been created in India to divert water from the Ganges to the Hooghly River. He said the problem wasn't the lack of fresh water flowing down, it's because of the lack of fresh water, the seawater was moving northwards. Second major problem is inland floods and droughts. This is affecting a lot of countries in Southeast Asia in particular, so that they're looking at what are called integrated water management systems. In China, for example, inland cities have to balance the problems posed by floods at some times of the year and droughts at the other. This is a common problem to 400 cities in China. So they've implemented what's called the Spring City Program. It's a pilot covering 16 cities, and it combines flood prevention, flood mitigation, and water retention schemes. And if it works, it will be rolled out to the other 400 or 380 odd cities in China nationwide. To To prevent flooding from storm surges and sea level rise, all countries are considering strengthening their coastal defences. That means putting in what are called hard engineering structures. In Singapore, for example, they're already elevating coastal roads, and they're putting in flood prevention schemes in the way of elevated seawalls and coastal protection structures. In Singapore, 70% of the coastline is already protected with some sort of coastal protection structure. So that only leaves 30% that they've got to cover. The other big problem, and this is very apparent in Hong Kong, is what I call waterway modification. To improve the channeling of waterways to remove floodwaters, they're armoring the lower reaches of river channels. I recently drove down a 5-kilometer stretch of road facing the mud flat in Deep Bay that SG has just talked about. Out of the 10 streams I've crossed, 10 were armored. Now that armoring is occurring where CR would normally spawn. So there are no riverbanks for them to spawn in. The final big issue is saline incursion. The upstream retention or diversion of waters affects downstream flow, and that leads to the inundation of seawater in the lower reaches. So this will actually serve to shift the spawning habitat of CR upstream. Climate change storm surges and rising sea levels are also causing saline incursion. And this is has been identified as a major problem in China. One of the reasons being that it's affecting the agriculture of coastal lowlands. So the government's response, or one of the government's response, is the use of alternative salt-resistant crops in coastal lowlands, such as salt-resistant rice. Well, if you want to grow rice, you have to build paddy fields, and paddy fields have embankments. So those embankments in some areas will be on the spawning and juvenile foraging habitats of CR. So the overall combination of upstream projects and rising sea levels is going to move CR spawning grounds into what I call unknown territory. I have no idea what's going to happen. So in summary, in terms of distribution, CR has a wide-ranging distribution characterized by small discrete populations. Now, it's difficult to know if these small discrete populations are a natural function of nature, or they represent relic populations struggling to survive. One of the previous presenters talked about long-lived species, and I really wonder if CR is a long-lived species which has developed a strategy which doesn't need to have so many such large populations as the other three species. That, that's a question for the scientists to involve, but I really do wonder why. The problem is, as well, we don't actually know what a viable population is for CR. The only thing I can say in terms of populations, scientists across its range are in agreement. The populations have declined and are declining. In terms of threats, it's faced with multiple cumulative threats, and all of its life stages are under threat: spawning, larval development, juvenile foraging and development, subadult and adult. There's extensive loss of spawning and juvenile foraging habitats throughout its range. It spawns on the high lunar tides. Artificial fisheries deploy their nets on high lunar tides. The subadults and adults are extensively caught as bycatch. Serious future threats are posed by climate change, and more importantly, the government responses to climate change. And because it inhabits estuaries and enclosed bays, marine incursions will push its spawning habitat into coastal defences, village areas, and waterway modification schemes. So the reality is there's nowhere for the spawning and juvenile development habitats to migrate to. The full impacts of saline incursion have yet to be thoroughly assessed. All I'd like to do is just a very short note on the likely red list assessment. Under the IUCN red list categories, we've got category A, which is population size reduction measured over the longer of 10 years or 3 generations based on any of 4 categories. And the 4th category are inferred, suspected population reductions. The causes of the reduction may have not ceased or may not be reversible. And the reality is that the causes of reduction for CR are continuing, and many of them are not reversible. So I'm not going to say what the assessment is going to be, but in terms of loss of habitat, for example, critically endangered would be 80%, endangered would be equal to or more than 50%, and vulnerable would be equal to or more than 30%. And that's really all I'd like to say. I'd just like to not only thank AJ for organizing this, but I'd like to thank most of the participants, because I've just used your material to brief you, and I couldn't compile this assessment without all of the help and the input that I've had from a lot of you guys. So thank you very much.
Thank you, Kevin, for your update on CR red list assessment. I would like to open the floor for the question to the last 2 presenters, SJ and Kevin. If there is any question, please unmute yourself and ask a question.
Can I say one thing to Jerome?
Yeah, he's there.
Yeah, look,
Yes,
no, look, Jerome, the genetics is critically important to these assessments. I mean, critically important. The way I describe these things to people is, it's like Photoshop. The more layers you have, the better picture you've got. And without the genetics, a lot of the population surveys wouldn't make sense.
Yeah, Kevin, I completely agree. I mean, I think in the future, if we can gather information from different places, Hong Kong and also other Asian places, I think we should include them in the assessment. I mean, and yeah, I really hope that. I mean, I'm very happy to contribute things that will be able to help your assessment.
The only thing I'm going to say as well is, one day, if you can explain to me what WMD means in simple terms.
Okay, let's go for lunch or dinner one day.
Yeah. Okay. But honestly, the genetics is integral to getting this right.
Okay, right.
Yeah.
All right. Okay.
Okay.
We'll do that.
Thank you, Jerome, and Kevin as well. Is there any other question? There is one question to Professor Paul Shin from the audience. May I ask if there is no question to Kevin and SG? Anyhow, the question from Ahmad Jalal Khan is thanks for sharing us your excellent info regarding the aquaculture activities of horseshoe crabs. Could you inform us which countries are now doing excellent in aquaculture of horseshoe crab during the polyculture technique? What are those fishes are cultured along with the horseshoe crab, it seems? Yeah,
I just saw the message. I just respond in the chat. To my knowledge, I think that China has been very good in terms of aquaculture for the breeding and culture of juvenile horseshoe crab. And also, I think that Malaysia is doing well as also because as Farida told me a couple of days ago, in Malaysia they have already established an aquaculture center or laboratory to breed and culture the juveniles. As to polyculture, I'm aware that the experiment that was conducted in Taiwan was based on mixed culture of juvenile horseshoe crab, the transparent horseshoe crab, with a baby or juvenile snail. Okay, the Babylonia or the baby law, which is the economical species that you can purchase in the market, and not fish. I'm also aware that Billy Kwan in Guangxi, China, is also doing a project with polyculture as well. I'm not sure what species he's using as the companion to juvenile horseshoe crab. But such kind of culture, polyculture, usually rely or require a pretty big area, preferably outdoor ponds. And so that's the information I have. So I don't think fish has been used as kind of companion species in the trial in polyculture yet for juvenile horseshoe crab.
Thank you, Prof.
Thank you for the question as well as the answer from Portion. Is there any other question from the floor? I have one question to Kevin regarding the CR update in IUCN release assessment. When possibly the results will be out in IUCN update?
Okay, I'm actually in full mode now writing. I would have to think that I'm halfway through doing it. I'm doing about 4 to 5 hours a day of typing. So the whole thing is based on typing. Once I finish that, it will then go to the review committee and they will take about 6 to 8 weeks. I think, I'm not sure if the deadline for submission this year is August or October. Maybe Paul or Mark can do it. But I would think the thing will be published next year. I mean, realistically, we submitted the TT one in 2018. It was published in 2019. The one thing I would say is, again, as with TT, when I started this, I just had no appreciation for the amount of data that was actually there. So, and one of the things I want to do is do justice to all your work, both yourself, Brian, all of the scientists across Southeast Asia. I will do that, but it's now reliant on my typing speed, and
okay,
I'm not going to tell you how bad that is.
I may add that to Kevin remarks is that I guess that the deadline for submission for the IUCN red list is sometime early September, something like that. Usually,
okay. Yeah.
Usually, they have the deadline around middle of August. But I think I received a message from Dave Smith. Probably Kevin, you may also have received it. I think that you should be able to find it on the IUCN website about the deadline.
Okay. Yeah. Yeah.
So in that case, if we will have to submit it in late August or September, then possibly we have to ready in the middle of August, the latest. That means if you back calculate and the review committee or the group that within the specialist group that have to review the information will take some time. Hopefully that won't be long. So again, there would be a couple of months, or 1.5 months, or 2 months for Kevin to get it done in the probably mid-July or the latest the end of July, so that we have enough time to go through the process. Mind you, because even though we submit the finalized document to IUCN, they had to go through another round of review within themselves, and they may come back with questions or comments that we had to revise our submission. And so that's why we take a bit more time before the assessment document is approved and we published it online. Probably next year, if we're following the schedule for the transparent horseshoe crab, that probably will be March or April next year. For all of the information, there are two application submission deadline for wet list assessment every year. One is around March, and the other is about August, end of August or September. So if we miss this round in late August or September, we have to wait to the next submission date, that will be March.
Thank you, Professor Paul, as well as Kevin. There is one update that I wanted to share with Kevin here is recently we have one paper accepted in evolutionary application, of course, with the National University of Singapore, Dr. Tang's group. It's on CR, different dispersal ability. It's again on genetics, but I will try to explain to you in simple terminology, maybe with Jerome. Thank you. Okay, if there is any other question from the floor, please.
Well, I've just got one question, which follows on from your question. I think when Paul was doing his presentation, Dr. Shin, I wrote down, are there any papers in publication or that have recently published that I need to know about for the CR assessment? And you've just mentioned one, but if anyone else has got anything that they're either in the process of getting published or has just been published, can they alert me to it?
I think there are a few other papers I have seen it as well.
Yeah.
So I will share with you, and if maybe the people can post in Horseshoe Crab Facebook group.
Yeah, that would be great.
Yeah, great. Thank you to all the presenters. I would consider the time limitations because of people are joining from different time zones. Really apologize if there is any technical glitch. We couldn't post it directly into YouTube live streaming because of some issues. By tomorrow, it will be posted and no one will miss any of the information. And with this, I would like to thank all the presenters and also all the participants for your immense contribution to this program. Without this program would not have been possible. Thank you very much. I would like to also thank the organizing committee. Behind the screen, there are two young guys, Mr. Anthony from IPB, as well as the three people, by the way, and there is one sister. For them also, there is big thanks. I would like to thank also all the participants, and whoever missed it, you can watch it in the YouTube live stream, YouTube very soon. Thank you very much once again, and have a nice day and stay safe. Thank you. Bye-bye. And there is a photo session. I'm so sorry for that. If possible, if everyone can open the the video, we can have a photo session. Mr. Anthony, I think you can take over.
Yes, sir. Thank you. We have two windows.
Okay.
I will take for Windows 1. 3, 2, 1, smile. Okay. For Windows 2, 3, 2, 1, smile. Thank you.
Thank you, everyone. Thanks a lot for joining.